Dear students, now we are going to discuss about PCR technique, process and the program. So PCR is one of the most important technique in modern scientific world. There are plenty of research problems are solved using PCR techniques. So PCR is nothing but a polymerase chain reaction. So PCR is an in vitro technique for the amplification of DNA at particular region. PCR amplification is achieved by using oligonucleotide primers. There are two different primers are needed for PCR technique. One is a forward primer, another one is a reverse primer. The forward primer is a one which attached with a forward strand, and the reverse primer is attached with a reverse strand. So this is the simple explanation about the PCR technique, how it works. So when you are applying a simple copy of DNA, so finally after the completion of all cycles, you will get more amount of DNA copies. So that is because of the polymerase enzyme and the primer. So for example, if you are adding one copy of DNA, you are running a PCR, and finally it will give you two copies of DNA sample. The two copies of DNA sample again undergo the PCR technique, it will give you the 8 copies of DNA. So finally it will give you more number of DNA. So that this is the reason why we need the PCR technique. So this shows how the DNAs are continuously multiplying in the PCR machine. The process wise, the PCR is divided into 3 different processes. One is denaturation, another one is annealing, last one is extension. So denaturation, in the case of first process, denaturation, the double stranded DNA will get into 2 single stranded DNA at the temperature of 94 degree centigrade. 94 degree centigrade, what will happen in the DNA is the hydrogen, all hydrogen bond present in the double stranded DNA will cut down. So finally we will get 2 single stranded DNA. The next step is the annealing. So annealing usually carried out around the 54 degree centigrade. It can be extended up to 60 degree centigrade. So what is happening in the annealing is the primer, whatever the type of primer we have added, the primer will attach with the 5' end of the template DNA. And the last step is extension. So in the case of extension, whatever the primer attached in the template strand, it will continue, the elongation process will continue for copying the DNA. This is happening with the help of DNTPs, and all the nucleotides are attached in the complementary strand by the enzyme called Taq polymerase. Taq polymerase is one of the most important enzyme used regularly in the PCR technique. So to run the PCR, we need to prepare a master mix. That is, the PCR master mix is nothing but all reagents are put together in a single vial, which is needed for the execution of a PCR. So the PCR reaction contains, the reaction mixer contains target DNA, 2 primers, that is forward primer and reverse primer. The length of the forward primer and the reverse primer should be in the range of 20 to 30 nucleotides length. And thermostable DNA polymerase, that is Taq polymerase, nucleotides DNTPs, and magnesium buffer, Taq polymerase buffer, and milk water. So these are the chemicals needed for the preparation of master mix. So simply the PCR technique includes the different temperature. One is 94 degree centigrade, that is the denaturation, then 40 to 60 degree centigrade, that is for annealing, and 72 for extension. So finally all these cycles are ended. Then the program will go to the storage temperature, that is usually we will keep it in the 4 degree centigrade. At the centigrade, the DNA will not denature, so it will be in a safer condition. So for successful running of PCR, we need to have a specific primer, because the primer are very important to elongate or copying the complementary strand. So selection of a primer is very important before starting the PCR technique. So we need to have a extra care when we are selecting primer, PCR primer. And the DNA polymerase. DNA, already I have told you that the DNA polymerase is very important because this is the enzyme which add nucleotide to the template strand as a complementary strand. For the synthesis of complementary strand for this template strand, the DNA polymerase is very important. The DNA polymerase here it has a unique feature that and this enzyme was derived from the special bacteria, that is Thermus aquaticus. This organism usually grow under extreme high temperature. Originally this enzyme was isolated from Thermus aquaticus from the national park, Yellowstone National Park. Generally, the TAF polymerase is generally called as thermoresistant enzymes. So finally, once the PCR reaction, the entire cycle is over, we need to check whether our DNA, so whatever the DNA applied or the particular gene we have concentrated, whether it is amplified or not successfully, we need to run the electrophoresis. Electrophoresis is the technique by which we can understand the result of PCR. So when you are running the electrophoresis along with the PCR product, you have to run DNA ladder. So you have to run it for 3 hours. For electrophoresis, usually 1% of agarose gel is recommended, and with the help of the ladder we can identify whether the particular gene are amplified or not. Even the gene is amplified, then you can separate the gel. You can separate the amplified region to the different the isolation buffer, and you can proceed for some other purpose like gene sequencing and the sequence analysis and further steps. So this is the one of the most important the technique in biotechnology, microbiology, gene technology, and biochemistry, and all the fields. So this is the application of PCR. The PCR is used to classify the organism at the molecular level. This can be used for genotyping, mutagenesis, mutation detection, and sequencing, cancer research, detection of pathogen, DNA fingerprinting, drug delivery, drug discovery, genetic matching, genetic engineering, prenatal diagnosis. This can be used for the molecular identification. So you can identify this particular species at molecular level. Detection of pathogen. You can detect the pathogenic gene, whether the pathogenic gene present in the particular pathogen or not. So this is the picture shows the PCR product, how the PCR product will appear in the electrophoresis. So this is about the PCR technique. This technique is used in a different field. Thank you.